we'll demystify CRISPR, how a bacterial immune system became a precise gene editing tool. In this brief deep dive, you'll learn the biological background, the mechanics of Cas9 and newer editors, key applications, and the main ethical and technical challenges. Chapter 1. Origins. CRISPR in bacteria. CRISPR's story begins in bacterial immune systems. In 1987, researchers identified unusual clustered repeats in E. coli, later named CRISPR, clustered regularly interspaced short palindromic repeats. Bacteria acquire short DNA fragments from invading phages and store them as spacers between repeats. When a familiar phage returns, CRISPR RNAs guide Cas proteins to recognize and cleave matching sequences. Key milestones include the 1987 discovery, mid-2000s recognition that spacers derive from viruses, and the transformative 2012 demonstration by Jennifer Doudna and Emmanuel Charpentier that Cas9 can be programmed with a guide RNA. By 2013, groups had adapted CRISPR-Cas9 for precise editing in mammalian cells, enabling biotechnology uses. Chapter 2. Mechanism. How CRISPR edits DNA. CRISPR editing uses Cas9 endonuclease guided by a 20-nucleotide RNA that pairs with a DNA target adjacent to a PAM. SP-Cas9 PAM equals NGG. Cas9 creates a double-strand break. The cell repairs it by non-homologous end joining, NHEJ, which often produces small insertions, deletions, or by homology-directed repair, HDR, if a donor template is provided. NHEJ is fast and dominant. HDR efficiencies in primary cells are often low, commonly 1 to 20%, while NHEJ yields higher mutation rates. To avoid double-strand breaks, researchers developed base editors and prime editors. Base editing can convert C to T or A to G with up to 150 to 80% efficiency in some cell types. And prime editing, 2019, programs small, precise changes with fewer indels. Chapter 3. Applications. Medicine, agriculture, ecology. CRISPR is already used across medicine, agriculture, and ecology. In medicine, ex vivo editing of hematopoietic stem cells, using CRISPR to reactivate fetal hemoglobin, has produced durable clinical benefit in early sickle cell and beta thalassemia trials, CTX001. Freeing some patients from transfusions. Researchers have edited T cells for cancer immunotherapy and used in vivo delivery in animal models for metabolic and retinal disorders. In agriculture, CRISPR produced a non-browning mushroom and disease-resistant crops. And regulators in some countries treat DNA-free edits differently from transgenic GMOs. In ecology, laboratory gene drive systems in Anopheles mosquitoes demonstrate the potential to suppress malaria vectors, but environmental and containment challenges remain before field use. Chapter 4. Challenges and implications. Despite promise, CRISPR faces technical and ethical challenges. Delivery into specific tissues remains a bottleneck. Viral vectors, AAV, can carry CRISPR components but face size limits and immune responses. Lipid nanoparticles offer alternatives but vary in efficiency. Off-target edits are reduced via high-fidelity Cas9 variants and thorough sequencing, but rare unintended mutations and chromosomal rearrangements remain concerns for in vivo use. Ethical issues include germline editing. Heritable changes were internationally condemned after the 2018 case of edited embryos, and equitable access. Advanced therapies cost hundreds of thousands of dollars. Governance frameworks, rigorous preclinical safety data, and international norms will shape which CRISPR applications proceed to widespread clinical or environmental use. CRISPR repurposes a bacterial immune system into a programmable DNA editing toolkit. It enables precise gene changes with broad applications in medicine, agriculture, and ecology, yet faces delivery, safety, and ethical hurdles. Responsible development, robust safety testing, and international governance will determine its societal impact.